Go for it, right? And welcome to another show. There you go.
Yeah.
That's a proper intro. There you go.
Right. Okay.
And who is it? Who is it that we have down here, Chris?
Yeah. Our special guest, Will, well known to our local group in the Northeast community, is familiar face to everyone. He goes every meeting I go to. He's always there. So an opportunity to have a bit more in-depth understanding about Will. So welcome, Will. Thank you for coming on the Amigos.
Oh, well, thank you for inviting me. I hope I don't disappoint you. Right.
Well, you'll disappoint both of our audience.
Yeah. All right. Yeah.
Right. So what have you been up to this week then, Will?
Will, to introduce himself and where he's coming from and his background.
Yeah. Okay.
Yeah.
Sorry.
Yeah. Okay. So William Hitt, like full name, right? And I have to say that I used to be a biomedical scientist because I pretty much packed it in last December, so I was a biomedical scientist in microbiology, so I worked in the NHS for over 30 years, in microbiology labs, so doing sort of testing, sort of mainly for bacterial infections, so doing culture and sensitivity testing.
This was at the RVI, was it, or the Royal Freeman?
No, actually, most of my career was at Queen Elizabeth Hospital in Gateshead, and then more recently I was working at North Tyneside Hospital from about 4 years ago until I packed in the job in December last year. So yeah.
Why did you pack it in?
Well, yes, that's a good question. Well, obviously, you'll have noticed that this so-called pandemic thing sort of kicked off I think it really properly sort of kicked off about 18 months ago now, isn't it? When they decided to lock us down,
Yeah.
And obviously, eventually, anyway, we got involved with some of the testing, the notorious sort of PCR tests. Interestingly, we weren't actually given the tests to do early on. When I don't know if you remember, there was daily press briefings that Matt Hancock did quite a few of them, and early on he was saying stuff like, "Oh well, our testing capacity in the whole of the UK was very low, that they could only do about 2,000 tests, in the whole country or something like a day or something like that," which was an utter nonsense because there's from what I gather, there's 130 pathology labs around the country in the NHS. So I mean, I know our lab, at the time with the analyzers we had and stuff like that, if we'd been given the test kits and the reagents, we could easily have from day one being able to do about 800 tests a day just in our lab, which is not a very big one, but as well, there's many bigger labs than us. So So if you multiply that by 130, you can see that we could have done many tests. If they'd involved the NHS labs, in like from the beginning. So
Well,
like, I mean,
someone who's got a lot of experience in this field who actually did research on it long before this kicked off was Kevin Corbett.
yes,
And which is someone we might be speaking to shortly, like in the near future.
Next week.
So, I mean, we all know.
Yeah, next week.
Well, I hope so. Yeah.
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 No, that would be good. Like, yeah.
And we know how ridiculous it is.
Yeah,
I mean, the way I always characterize the PCR test. I mean, you tell me if I'm wrong, Will, when I say this, right?
Yeah.
But it's like I often characterize it as being like if you put down all the notes of, like, say, Beethoven's Fifth Symphony, or da 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 da, and you put them all down one after the other, and then you selectively chose one little part of it. Let's say G, D, E. So you look G, D, E. Now you get all the other pieces of music and you lie all them down, and you look for G, D, E in them. You're going to find it everywhere. You're going to find it in Dolly Parton records. You're going to find it in classical music. You're going to find it in folk songs and everything.
Yeah.
And then people are going to point on it and go, oh, look, there's Beethoven's Fifth Symphony. And it ain't,
Yeah.
So all it is just like sort of very, very minimal associations with it. And then they're trying to say that this is proven. That it's something that, let's be honest, hasn't even been isolated in the first place anyway. So, that's a lot of crap,
Yeah, that's right. No, that's a really, really good analogy, actually. Yeah, like because you are just looking for a very, very small or maybe 2 or 3 very, very small sections of an alleged like viral genome. So like the viral genomes, like obviously they're made up of what you call nucleotides, like so that's what you say. Each one's like a letter, like and this SARS coronavirus 2 is supposed to have about 30,000 of these letters, and the PCR will look for a small section which will be around about 200 letters long, and
when you consider that we share our genetic material with a banana by about 62%, I mean, it's like,
I think it's much more than that, actually. I think we've got,
that right? Is it
right. Maybe more, maybe like 80% or something like that, but I'm not sure about that. I'll really look that up.
It gives an idea anyway as to ridiculous.
Yeah, that's right.
So, and I'll tell you another thing as well, like sort of when they're talking about these variants, again, like, correct us if I'm wrong, but as far as I'm aware, in modern scientific understanding, if you have a resistance to a virus, and you encounter another virus with a 70% similarity to it, you will still have a similarity to that virus if it's 70% similar. And these variants only vary by about 1% from the one that they're supposed to have been injected against. So even according to their science, it's bollocks.
That's right.
It's crazy.
Yeah, I think Mike Yeadon, like, obviously has talked about this, and he reckons that people who were infected with the SARS, the first SARS virus, like 17 years ago, like actually had immunity to the new SARS-CoV-2 virus, like and I think they're only supposed to be about 80% like similar, like so these variants are only like these variants, if you believe them like to be real, like are only supposed to be like about 1% difference, like or maybe like 3% at the most.
Yeah, I mean, that's the crux of it, isn't it? If you believe it to be real, kind of it's almost as if you're embellishing a fantasy. As if someone's told a fairy story, and we're embellishing on this fairy story, like sort of as if we're talking about Star Wars being real, and then asking, well, does Darth Vader really like football? Like sort of, is Princess Leia really related to somebody? It's almost given it validity when you think, hang on a second, let's just have a rewind here, like, you know. And
that's right.
Absolutely
I mean,
crazy.
It's as you said, like the virus has never been isolated, and I mean, and this is actually true for all alleged pathogenic viruses. Never ever been isolated and properly sort of characterized, and then never been like shown like that the
Isn't it
virus
true?
can cause disease on its own. Like
and isn't it also true that I think it was back in 2010 where Robert Kennedy Jr. won a Supreme Court case where it was ruled, where the judge ruled, no vaccination has ever been tested for safety, ever. So when they come out with all these things about like sort of it takes 7 to 10 years, it's again, it's embellishing this fantasy in the first place. There is no safety.
Yes, they'd never do any like safety tests against like like some sort of like an inert placebo or something like that. They whenever they test the new vaccines, it's always against an old vaccine.
Is this the Cox postulates? Is it?
No, I'm just saying like when they do like safety testing for like when they do like the sort of like clinical trials for new vaccines, like they'll actually compare them to like old vaccines or what they call adjuvants, the stuff that they add into the vaccines to actually generate a stronger immune response, like
Is this how they get the excuse for putting in things like mercury and tungsten
and aluminium and like all kinds of like toxins like that, so basically they're comparing one like toxic substance to another toxic substance, and then they say, oh well, they're equally like and then
Kelsaprice,
and then they sort of like say, oh well, it's safe because it's actually like there's no more harm in this one than in the placebo, which was
honestly, it's like when they have if there was a thermometer was broken at a factory, they would have to like evacuate the factory and get the hazmat team in to clear it before people could go back in, and they're injecting these things into our children,
Well, I believe the question's been asked to these vaccine companies: have they ever done a true double-blind study with a totally inert vaccine? And their reply was to say no, because it would be unethical to deny that person the vaccine in the first place. Such a load of nonsense, but that's what they say.
So
this harks back to the Cox postulates, doesn't it? Where they've got like is it 4 criteria they have to fulfill, and it doesn't matter whether it's yellow fever or whatever it is. In order to basically be recognized as a pathogen, it has to qualify through these 4 or 5 criteria. If it fulfills 3, it's not enough. It has to fulfill all 4. This thing hasn't fulfilled 1. Largely due to the fact that it hasn't been isolated.
I mean, So I can quickly run through like Cox postulates for you. So like this is how you're supposed to prove that a microbe is an actual pathogen. Right? So like the first one states that you should be able to find this microbe like in abundance, like in an infected like individual, like and then you should not be able to find it in abundance in a healthy individual, like so then you should the second one is then that you should be able to isolate this microbe, like and get like a pure culture of it. Like this was kind of like more to do with bacteria, but you should still be able to do this with like viruses, like but and then when you've like got your isolates, like this pure culture of like your microbe, you should then be able to inoculate a healthy individual with this like isolate, and then that should be able to cause the same disease in that healthy individual, like, and then you should then be able to see again this microbe like in abundance in this now infected individual, and you should again be able to isolate it from that individual, and then prove that it was the same microbe that caused the disease in the first like infected individual, like, you know. So that's basically. So I mean, like, you can see the logic there. Like, you know, without all that, you haven't proved that this organism actually causes a disease. Like, you know, just because, for instance, if you just saw this like organism like in someone with a disease, that isn't proof that that organism's actually caused it, has it? Like, you know, so then, like, obviously if you can isolate it, and then you can actually inoculate someone who's healthy and hasn't sort of got the disease, and then it does actually cause them disease, like after you've done that, like, and then you're able to like then you know, isolate that same organism again from now this newly infected individual, then that, you know, that is pretty good sort of proof that organism has probably caused, yeah.
As I understand it, Will, the PCR test only tests for the presence. It could even just find one single strand, and then it's positive. It doesn't look at the quantity or the load of this so-called virus or germ. Is that right?
Well, also what it does is it amplifies it over and over again. So I mean, it just
yeah. So anyway, like
because it's only one little sample. It's only one little sample. It doesn't say the quantity of this virus present in you.
Not even. I wouldn't even say it's even a quantity of that, because all you're taking is a genetic marker. Yeah. So it's like sort of, it could be, you know, like again when we're talking about the how it's related to a banana, you could be picking up traces of the banana that had that same genetic sequence in it. You know, who says it's a you know, like a deadly pathogen that's come from outside.
So if there's one rogue broken little fragment of some old antibody that's been there for 6 months, and the PCR just happens to come across it, you get a positive test. Is that right?
So let's go back to, like, you know, so I'm talking about Koch's postulates, and like obviously, like, so a key key thing is to like isolate your microbe in the first place, right? So like and what Charlie's already mentioned is that we've never isolated this alleged virus, and like, you know, like we've never isolated any alleged virus, right? So like, if you've never isolated the virus, you know, how can you actually have sequenced its genome, like, you know, and
how can you produce an antidote or a vaccine for it?
Exactly.
What you're vaccinating against, you know.
And then if you've never isolated that genome, like, and sequenced it properly, how can you actually have a PCR test that's looking for a small section of that genome, like, you know, that a small section, you know, that belongs to that genome, when you've never actually isolated it, like, you know.
Fantasy based on a fantasy.
It is. It's a, you know, it's.
So I'll tell you what. A straw poll. Who thinks this is a load of bollocks? Carried.
Yeah.
we are a very small spectrum of the general public.
But
we've got Will here. We'll take his views as
that's unanimous then.
That's unanimous. So it's working out how to deal with this fantasy, isn't it? And the people who are trying to impose it.
Yeah, Will, just on the topic of this SARS-CoV-2 virus, right? From what I've seen on videos and doctors saying, it doesn't actually really exist. It only well, what they're injecting to it, certainly the spike thing that they're injecting, it only exists in situ. Is it? No,
in silico.
Yes.
In silico is the term they use, which means it's basically only exists in a computer program, like, you know, because like it's again down to like this, you know, the way that virologists like claim to actually isolate viruses. Like, you know, they you know, it's not isolation by anybody else's definition. Like, you know, like, I mean, as a like more of a bacteriologist is what I was doing. Like, we, you know, I can say for sure that we isolated bacteria. Like we did that like hundreds of times every day. Like, you know, because we can just grow them on like petri dishes, and like they'll grow into nice separate colonies. Like, and you can pick off the different colonies. Like, you know, so and then, you know, do a separate culture from that, and like you'd have yourself a nice pure culture, like of your isolated bacteria, like, you know, that's, you know, contains only that bacterium, like, you know, basically, like, you know. But like with viruses, they've never done anything like equivalent to that. It's like, you know,
Chris mentioned there as well about the spike protein. Will, could you put in layman's terms, because we know that a lot of particularly women seem to be suffering from symptoms who are associated with people who have been double vaxxed. And the thing that was said that was causing it was the spike protein production. Could you just elaborate on that in a little bit of layman's terms so people can understand how that mechanism works?
Well, this spike protein like appears to be basically the toxin like that causes the, you know, the serious symptoms of you know, so-called COVID. Like, you know, it causes like the blood clotting, like, and you know, it causes all the inflammation in different organs, like, you know, the serious sort of inflammation.
Because in when DNA, it's kind of like a blueprint, and it codes for a protein, doesn't it? So that basically what it's doing is it tells the ribosome to code for a protein. So everything that we're made of, it doesn't matter whether it's bone or teeth or whatever, it's a protein of some kind. So this spike protein is what it's doing. Is it creating something that's surplus to requirements, shall we say?
No, what you know, what these so-called vaccines, you know, these really gene therapy like agents do is like, you know, depending on which one, like with the Pfizer, BioNTech, and Moderna, it's got mRNA, like messenger RNA, which is like, as you were alluding to, that's what goes to the ribosomes allegedly, like, and where, which is like the factories where the proteins are made, like, you know, and this and the mRNA is like a blueprint for a protein, like, you know,
that's what we always say. It's like a factory with a blueprint.
And so this mRNA goes to the ribosome, and then it's you know, the proteins, the ribosomes make the spike protein. Then, like, you know, like, because you know, they're supposed to just like be like factories, and they'll just like make whatever codes like they're given, like they'll make that protein, like, you know, so that's.
So why do they call it a spike protein? Is there
something
that's
relative because
to that
allegedly it's you know, when you see these electron micrographs of the alleged virus, it's like a little ball with lots of little spikes sticking off it, like, you know, and this is actually supposed to be the protein that forms the spikes, like, so that's why it's called the spike protein, like, you know, So it's around the outside of the virus.
So it's like it's the first thing that would come into contact with other proteins or viruses. Yeah. Right.
Yeah. So that would come into contact with the cells, and like apparently, it's sort of like it'll bind to what they call the ACE2 receptors, like in like and those seem to be present in quite a lot of different tissues, and and they're so
are you saying this spike protein is not unique to COVID?
Well, the thing is, it's actually been patented. Back in about 2002 or something like that. It's like when actually before the first SARS, before the first SARS virus, and so that sort of suggests that was kind of an engineered thing as well. The first SARS outbreak as well, because then
there's also a lot of connections with HIV as well, isn't there? Which again Fauci was in charge of, and anybody who knows the debacle around Judy Mikovits, who was one of the research directors under Fauci. And there was all kinds of stories where they said that isolated the HIV virus, and then Fauci deliberately put it off so that they could patent it for the US pharma industry and things like that.